Part B of chemistry. Hoping you're not thinking chemistry is that bad in this class. Remember, this is a biology class. It's not a chemistry class. So we're just introducing some things to you. Part A introduced the fact that the body is made up of a bunch of chemicals, and those chemicals react with each other. That's physiology. Okay. We introduced basic chemistry. So matter and energy cannot be created or destroyed. They just get kind of shuffled around to be maybe a different kind of energy or just used in a different location. And then the molecules that we have get broken down or built up into different sort of molecules. The whole idea, ashes to ashes, dust to dust, but then we can build up new beings by, the decomposers break things down, but then life creates new life. We use the decomposed material to make new life. So those elements are just being recycled constantly. One thing I didn't mention in the PowerPoint the other day is that those elements, they came from the stars. So all of the things that we have on Earth have been broken off of little pieces of stars. Isn't that cool? That's so cool. All right, I digress. Okay, so matter is something that takes up space, whether it's gas, liquid, solid. It's something that takes up space. Even if it's a molecule you can't see, it's there. Energy is the capacity to do work, and it's what moves matter. It's what creates the chemical reactions. And there's different kind of energies. You have the kind that's stored, just waiting to be used, and then you have the percentage of energy that's in motion. That's kinetic energy. Forms of energy, we went through that. Chemical, electrical, mechanical, radiant / electromagnetic / heat. Then we talked about the elements. So the periodic table, oxygen, carbon, nitrogen, hydrogen, those are the biggies in the body. There's a whole bunch of them that we will never mention in this class because it's a biology class. But carbon, oxygen, hydrogen, nitrogen make up almost all of the body, 96%. When we're talking about the element, a single piece of that element is an atom, and it's made up of different parts. Just like we're made up of different systems, an atom is made up of different parts. The center of the atom is made of a nucleus that has protons and neutrons. The protons are positively charged, and the sum of the mass of the protons and neutrons is the atomic mass, the mass number. Electrons on that piece of paper are going to be equal to the number of protons. That might not be how that atom is happy in real life. And we went into that, but I'll get to that in just a minute. But on that piece of paper, when we're writing down the subatomic particles, the protons and the electrons match up in number. The protons are always going to be the atomic number. So for example, carbon has 6 protons, so the atomic number for carbon is 6. Oxygen has 8 protons, so the atomic number for oxygen is 8. You change the number of protons, you have completely changed the element. Neutrons, so we'll say that we have carbon, and the most common form of carbon is C12 because it has 6 protons and 6 neutrons. 6 plus 6 is 12. So it's called C12. But we have things called isotopes that occur naturally, where an isotope is the same element, but we've changed the number of neutrons. Remember, if you change the number of protons, you've changed the element. But you can have isotopes that have different number of neutrons. So for carbon dating, that kind of carbon actually has 2 more neutrons. It has 8 neutrons. So it's called C14. You've probably heard of C14, especially if you love National Geographic and Discovery. Some of those isotopes might be radioactive, and radioactivity may or may not be damaging. We can use it in the medical field for medicine sometimes, or sometimes it's what caused a cancer. That's a different class. If you put some atoms together and you bind them, right, then you have stored energy in the bond and you've made a molecule. And in general, once you put a bunch of atoms, your textbook says more than 2 atoms together, then you're getting a larger molecule and it's going to be called a compound. When we put matter together and they don't bond, that's called a mixture. So we had different kind of mixtures, a solution, a colloid, and a suspension. I'm not going to ramble on about that. Look those up. You should know what they mean. And then for chemical bonds, that's the relationship of how these guys interact. And that has to do with the type of matter it is. Is it organic matter or is it inorganic matter? The inorganic matters usually are going to bond by ionic bonding. So recall how that happens. It's like dating, and they're attracted to each other. So that's usually going to be salts, acids, bases. And then the other kind of bonding that is a strong bond, ionic is strong, and so is covalent bonding. That's how organic matter bonds. Instead of dishing out his electrons to somebody else, with covalent bonding, they share electrons. They get married. Their rings get married together. What else do I want to say? Ionic bonds, so an ion is a charged particle, and if you lose an electron, you lost a negative, so it becomes a positively charged particle. That'd be a cation. If it gains an electron or 2, then it gained negativity, and that's going to be called an anion. Covalent bonds come in 2 different kinds. It comes in polar and nonpolar. Please review those. Remember, nonpolar is hydrophobic. Polar is hydrophilic. Hydrogen bonds is a weak bond, and what happens with that one is when we have polar molecules, that means it's an unequal pull on the electrons. So when you actually look at the physical structure of that molecule, one side of it, the electrons are hanging out on one side of the molecule more than the other. So that means that that molecule is a little bit more negative on one side, a little bit more positive on another side. So when it meets up with another polar molecule, that partial positive on one molecule is attracted to the partial negative on another molecule. It's a very weak bond. It's the reason you can easily break up water and put water back together. Chemical reactions, the equation is what you see, something plus something yields something. The things that you're adding together are called the reactants, and the final end result is the product. Sometimes you build things up. That would be, building things up is an anabolic process, and that's synthetic, so synthesis reaction. And what happens whenever you build things together, you bond them together, the energy is stored in the bond. So that's endergonic. A breaking things up is catabolic. So you broke a bond. When you break a bond, you release energy. So that's a decomposition reaction, and it's also an exergonic reaction. You decompose something, you broke it up, catabolism, and when you break bonds, you release energy. So that's exergonic. Finally, in biology, things that make chemical reactions go faster. Well, it depends on temperature. It depends on if things are hotter, that reaction's going to happen faster. If there's more concentration of a reaction, it's probably going to happen faster. If it's a tinier particle, it's going to move faster because it doesn't weigh as much. And then if there's a catalyst that helps things along, then it's going to happen faster. In biology, the catalyst is always an enzyme. Okay, so that's part A. Part B, biochemistry, is how these chemical compositions react in the body. So I've actually already mentioned this in part A. Inorganic compounds are salts, acids, and bases, and they do ionic bonding. And I'm going to throw water in there too. Water is inorganic. Almost always, inorganic compounds don't have carbon. The exception is carbon monoxide and carbon dioxide. Organic compounds are the things that we think about in our diet. Carbohydrates, fats, proteins, but also I'm going to throw in nucleic acids. So you've probably heard of DNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. In general, these organic compounds are what was I going to say? Organic compounds are covalently bound. That's what I was going to say, covalently bound. But you have to, in biology, you have to have both inorganic and organic to stay alive. Okay, water is this crazy important compound in life. It is very strong. By itself, a little water molecule or two isn't a strong bond, but you put thousands of them together, and 2 inches of water can carry off a truck in a flood. That's quite strong. And water will find its way through the tiniest little crack in a pipe. It is a very, very tight and strong bond when it's in multiple numbers. Here's a couple of other extra cool things about water. It has a high heat capacity. What does that mean? Well, it resists changing the temperature to itself despite what the temperature is around it. So it's 105 degrees outside, and you can go into the ocean. The ocean's not 105 degrees. Us personally, it's 105 degrees outside. Our blood is mostly water, but our blood's not boiling. So it resists those external temperatures around them, and it has a tendency to have very little temperature change to itself, despite how hot it is around it. High heat of vaporization, I'm going to allude this to perspiration. So we're hot, and the brain says, one of the homeostatic mechanisms is to sweat. Brain tells your sweat glands you need to put out some sweat. Why? Well, because that sweat is going to pull heat from our body, and it's going to use that to change from a sweat particle out into a water into the air. It takes a large amount of heat for that to do it, but it's pulling it away from us to help cool us off. Polar solvent properties. So water dissolves ionic substances quite easy. It doesn't bond to them, but little water molecules surround like say salt water, it surrounds the sodium, the Na pluses. It surrounds the chloride, the Cl negatives. So it doesn't bind to them, but it dissolves them and breaks those ionic bonds apart. It also forms water layers, hydration layers around large charged particles, which is helpful when proteins are floating around in our blood. Water is, if we don't have enough water in our body, we are going to die, because he helps bring whatever our cells need. It is the reason that we can carry things around in our blood. You must have water for so many of the reactions in our body. Decomposition. Add some water to it. That's hydrolysis. You used water to break something up. If you want to bind things together, take some water out. Take out an H2O. That's a dehydration reaction where we've made something, synthesis, by taking out a water. And then also it cushions us. So those serous membranes that we talked about, a large majority of the serous fluid is water, and it helps cushion things. And what about the baby that's in the amniotic sac with all that amniotic fluid? We are mostly water. The amniotic fluid is mostly water, and it cushions the baby, cushions our brain. We've got it around our brain. This is a picture showing how water dissociates salt. It's not binding it, it's surrounding the sodium, surrounding the chloride. Okay, moving on. So that was water as an inorganic compound. Now we're going to have salts as an inorganic compound. Salts are ionically bound. So they're going to be broken up into cations and anions. Those ions are often called electrolytes. You've probably heard of that term. Gatorade, Powerade, all those things. A new one I was introduced to is electrolyte, which is loaded with potassium if you're having cramp problems, or eat some bananas. They get that name because those cations and anions can create electricity. And we'll see that in lab. We must have so many of these ions to think, to move our muscles, to clot our blood, to do so many different things. If you don't have these things in good number, obviously you are out of homeostasis and you sure could die. If you get too much of them, same thing, you could die. There has to be a balance. Listed here some common salts in the body, not a test question, but sodium chloride's a huge one, yeah. Okay, so we've done water, we have done salts, acids, and bases. They also break up into particles, ions, and they also can be electrolytes. You can have strong acids or weak acids, okay? But let's start with acids are proton donors. That means when they break up, they're broken into a hydrogen ion and something else. Bases, when they break up, they release hydroxyl ions, which are OH negatives, and something else. Well, OH negative sure would like to meet an H positive, wouldn't it? Because then we have water. And what is the most important substance in our body? Water. How efficient if some sort of molecule in our body broke up and we had an OH hanging out or an H hanging out, how efficient would it be to put those two guys together to have more water in our body? Every millisecond of your life, this is going on. So that OH negative, because he is dying for a H positive, he's called a proton acceptor. Remember, hydrogen just has one proton. And in real life, he doesn't even have an electron because that valence shell only had one electron. So he likes to just donate that to somebody else. So now hydrogen in real life likes to float around as H positive, just a proton. So OH negative, also known as hydroxyl, is dying to meet H positives, protons. So they bases are called proton acceptors. Picture, please review it with your time frame. Okay, so you've probably heard of pH. pH, essentially what we're doing is measuring the amount of hydrogen ions in something. Okay, so bases and acids are put on a scale, and the more hydrogen ions that are in a solution, the more acidic it is. This pH scale goes from 0 to 14. So if I'm saying the more hydrogen ions that are in there, the more acidic it is, I'm talking about starting at the bottom, the 0, 1, 2. That side of the scale from 0 to 114 is the acidic side. Going up to number 14, that's very basic. Okay, so memorize that, and then I'll show you how, well, why is that low number? You're saying it's high in hydrogen ions. I'll show you. Looky here, when we have a pH of 7 in the middle of the page. That's neutral, by the way. Of course, between 0 and 14, 7 is neutral. And that's what pure H2O is. Distilled water is 7. So it's not acidic, it's not basic, it's neutral, it's 7. Look at how it's written down mathematically. 10 to the negative 7. So the concentration of the hydrogen ions is 10 to the negative 7. The 7 is the pH. Okay, so I'm telling you acid, 10 to the negative 1. That's pH of 1. If you remember from math, the negative 7 means moving the decimal over to the left. So 10 to the negative 7 would be moving the decimal from 1 to 0.1. If it was 10 to the negative 3, you would be moving a decimal over from 1 to 0.001. So write that down. 10 to the negative 1, you write it down a 1, you move the decimal over 1. So it's 0.1. 10 to the negative 3 would be moving the decimal over 3. So 1, move the decimal over 3, be 0.001. And I'm trusting that you're writing this down. Now look at your numbers. 0.1 and 0.001. Let's think about money. Is 0.10, is that 10 cents? 0.001, what kind of cents is that? That's not even a penny. Which one is worth more money? The 0.1. The concentration of 0.1 is higher than 0.03. So we're concentrated with more hydrogen ions at 10 to the negative 1 than we are concentrated with hydrogen ions at 10 to the negative 3. At let's say a pH of 14, so 10 to the negative 14, write this down, write down a 1, and now move the decimal place over a 14. How much money is that? That's not very much money. So therefore, that's not very much hydrogen ions. That's not a high concentration of ions. So the more hydrogen ions you have, the more acidic it is. If we're at 14, that's not very many hydrogen ions. So therefore, it's not basic. I mean, it's not acidic, sorry. It's not acidic. That is very, 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 very basic. Examples of acidity and alkalinity is the other word for basic. There's examples. Notice blood is at 7.4. So what is our blood? 7's neutral. So therefore our blood on average is a little bit basic. In the body, we have some acids, we have some base. Some of them are strong, some of them are weak. But when we have a strong acid like hydrochloric acid in our stomach, okay, it's fine for the stomach because the stomach is extremely well protected with mucus. But our intestines aren't. Our intestines aren't loaded with mucus. So when that foodstuffs that moves from the stomach into the duodenum, when that happens, we need some sort of buffer to keep our small intestines from being burned up from that hydrochloric acid. What a buffer is going to do is going to change a strong acid to a weak one, or a strong base to a weak base. And our pancreas actually puts out a buffer to protect our small intestines from getting all burned up. This is probably a little bit more information than you want to know. These are the different buffering systems. But the one that I just mentioned with the pancreas is the bicarbonate buffer system. Bicarbonate is going to bind with hydrochloric acid to make it weak. More examples. Please read this at your leisure. Those catalysts that I talk about that make chemical reactions happen faster, they need a perfect pH range. Enzymes are protein. Our body cannot go out of a certain range or else we will die. So when people are really, really, really sick and they start spiraling downward with their health, let's say at a hospital, they seem stable and now tomorrow they're worse and then the next day, wow, they're worse. Guaranteed their pH is getting messed up. Probably becoming more acidic, but the person that is crashing and burning, usually along with whatever else is messed up, so is their pH. And all the proteins in our body just start to denature and break up. Organic. Organic molecules are made of carbon, and the exception that I've already mentioned is carbon monoxide and carbon dioxide. Those are inorganic. They're not something that we consider as organic matter. Organic matter are the things that you think of as our foods: carbs, lipids, proteins, and then those DNAs and RNAs inside of whatever we ate. Yes? Because it's organic, it's covalently bound. So it shares electrons. They're married. Those valence shells bind together. There's none of this, here you can have this and you can have this. We're getting married. Already mentioned this, but the dehydration and the hydrolysis. You can take covalent bonds and add some water and break it up. That's hydrolysis. Lysis is to break. Or you can take organic compounds and take a water out of it, and then you have bound them together. So it's a synthesis reaction by dehydration. You took a water out. Organic compounds in general are very large, so they're polymers is what they're called, but you can break it down into something super, super tiny. So I've probably already alluded to this, but carbs break down into sugar. That's their tiniest building block, is glucose, for example. That's why people are scared of carbs, right? Because too much sugar is going to get eventually stored as fat. So the monomer of a carbohydrate is sugar. I use that example because most people understand that. This is just showing a picture. A polysaccharide is going to be, for our foods, is going to be a carbohydrate. So the smallest, the monomer of a carbohydrate is a sugar. Well, if you bind two monosaccharides together, two sugars together, then you have a disaccharide. You put a whole bunch of them together, they're a polysaccharide, also known as a carb. This is a cool picture showing dehydration and hydrolysis. I already told you to lyse something is to break it apart. So this bottom reaction is showing you are adding some water to those molecules, and they're separating those molecules. They're lysing those molecules thanks to water. Hydro is water, lysis break up, hydrolysis. Dehydration reaction, or making something, synthesis, you're taking a water out. You've just bound those guys together. So you have created a bond between both of those molecules. Endergonic reaction, synthesis reaction, dehydration reaction. It's all three of those kind of reactions. So carbs are actually the human's greatest energy source. Carbs have carbons, hydrogens, and oxygens in a 2-to-1 ratio, or hydrogen and oxygen in a 2-to-1 ratio. Carbon is the skeleton. And there's different classes of carbs. So we have monosaccharides, the single sugars, disaccharides, I already showed you that slide, 2 sugars, polysaccharides, many sugars. So here's that formula for a monosaccharide, CH2O in parentheses and then an N. So if I made, so a CH2O, if I made the N a 6, that'd be C6H12O6. If there's not a number in front of something, you assume it's 1. So we assume in this right here, pointer, this is C1H2O1. Make this a 6. 1 times 6 is 6. 2 times 6 is 12. 1 times 6 is 6. C6H12O6 is a glucose. Right there. Because it's C6, it's known as a hexose sugar. We have other hexose sugars too, fructose and galactose. And when we do DNA and RNA, you're going to meet the pentose sugars. So instead of having 6 carbs, they have 5. Remember that. Already said all that. So polysaccharides are what we think of in our food as starches. Okay? You can break starches down into sugars. There's 2 other kinds of polysaccharides that I want to, or carbs, that I want to introduce you to. Cellulose, you know this. When you hear about like, why don't we eat the grass? Well, because we can't digest it. Well, as long as it's not poisonous, you sure can. But you're not going to be able to use it for food. However, we're going to use the fiber from those plants. That's cellulose. That is a carb. It is very important for our heart and our colon health. So although cellulose is a carbohydrate, it's not one that we can absorb into our bloodstream. It passes all the way through our gut, but it's still very healthy. Also, some vegetables, of course, have a large amount of fiber in them in the form of cellulose. You can get the vitamins out of those vegetables. And actually, there's a little bit of sugar in some of them, like carrots. But they're very important for our heart and for our colon. Finally, the last carb I want you to meet, polysaccharide is a carb. The last carb I want you to meet is glycogen. Those 12 Snickers bars that you had because you're all stressed out, you're never going to use all of that sugar in one sitting unless you haven't eaten in days. But what your liver is going to do is going to take any extra sugar floating around and bundle it into a new molecule called glycogen and store that sugar as this molecule, glycogen. Then when you go on a starvation diet, your liver starts using the glycogen. It releases it into the bloodstream as glucose. Lipids, these guys are nonpolar, right? Oil and water. Lipids are like an oil. And the one that we think about in our diet is triglycerides. But there's a couple of others that I'm going to introduce today. I'm leaving out eicosanoids. That's why it's so small. So as I said, triglycerides are the lipids that we think of that are in our diet. But they can be broken down as triglycerides. That is a polymer, many molecules. They're going to be broken down into some monomers as fatty acids and glycerol. Triglycerides are 3 fatty acids and a glycerol. So think about this. This is saying they get triglycerides, our fats in our diet, get broken down into 3 fatty acids and a glycerol to their monomers. The way that they would do that is hydrolysis. You drink some water and you exercise, you're going to use that water to break those fats apart. Pretty cool, huh? Drink more water. Makes me realize that my water cup is empty. So the main function of triglycerides is for energy storage, but we also use fats for insulation and protection. This is showing, you will never have to draw something like this for me, but this is showing a fatty acid. This is a single fatty acid and a glycerol molecule. And the way that they're going to form is you're going to have 3 fatty acids and 1 glycerol. You're going to take out some water. So dehydrate it, and then you have fat. Here we go. There's those 3. This is a full triglyceride.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules.
You've heard of saturated fats and unsaturated fats. A saturated fat means that the carbons that are in that molecule are saturated with hydrogen, single covalent bonds of hydrogen. Hydrogens are making up every link to that carbon that's possible. Saturated fatty acids are solid at room temperature. Unsaturated fatty acids means that it's not every bond is not taken up by a hydrogen. Sometimes there'll be double bonds between the carbons. And when you put that double bond in there, it causes the fatty acid to kink and can't pack together closely. So therefore, instead of being a solid, it's a liquid. Omega-3 fatty acids are very heart-healthy unsaturated fats. Trans fats, those are not healthy. Why? Well, somebody has taken a saturated fat and chemically altered it. How have they done that? They have moved hydrogens around in a laboratory that have forced what was a solid state, saturated, to become a little bit softer by moving hydrogens around on the location of the molecule. So in a nutshell, it got moved, they moved the hydrogens from a cis location, CIS, to a trans location. You don't need to know what those mean, but that's how it gets its name. The hydrogen has been altered in a laboratory to be on the trans location of the molecule, and so trans fat, not healthy. Margarine. Okay, now this guy, you're fixing to get tired of hearing about this. You're going to hear about this to the ends of AMP2, so it's important. Another kind of a lipid, besides the triglycerides that you eat, are phospholipids. That cell membrane that I keep talking about that has rules on how you get in and out of a cell, Those cell membranes are made up of a whole bunch of these guys. This picture actually is showing how they look on a cell membrane. So instead of having 3 fatty acids, it only has 2 fatty acids. This is one of them, and this is another one. Plus, it has a phosphorus group. This guy right here is a phosphorus group. So up here, this is a bunch of PO4s. This is a bunch of PO4s, phosphate group, actually, phosphorus-containing group. And then these are the fatty acids. Okay, fatty, hydrophobic. But these guys, these little phosphate groups, are hydrophilic. That's why as the blood or tissue, whatever that's passing by that cell, its blood is made of water, right? It's all good. They're happy. Hi there. Hi, how are you? Hi. But it can't sneak into that cell really easily because of these guys right here. These guys are not water lovers. These are hydrophobic because they're fats. Oil and water don't mix. There's a picture of them. All the chemical structure. You will never have to draw that for me. Okay, steroid base has 4 ring-type structures. And the rings look a little bit different. Some of them are hexagons, some of them are pentagons. I'll show you that in a second. The most important steroid in our body is cholesterol. Why? The liver makes a lot of the cholesterol that's in our body. So yes, we eat it in animal products, but our liver also makes it because cholesterol is an important building block for some of the vitamins and for some of our hormones, and also for our bile salts. Bile is going to help us break down our fats that we eat. So cholesterol is actually a good molecule most of the time. Also, sometimes we're going to embed some cholesterol into those cell membranes, so we're going to squeeze them in between some of those phospholipids that I just talked about because it helps maintain the integrity of that cell membrane, helps make it strong. There's the stop sign house that was alluded to, when in fact stop signs are 8-sided, but that's what a lot of the other professors refer to it. So if you get one of the other professors for A&P2, I want you to know what we're talking about when we say stop sign house. So this is a hexagon, this is a hexagon, this is a hexagon, this is a pentagon. This is the base for cholesterol, for a lot of steroids. Okay, moving on to the next category is proteins. Proteins are in massive amounts in our body. Various kinds, various structures, but we need it in everything. And as a matter of fact, if you're taking a test and you're just absolutely not sure what the answer is, but protein is a choice, go ahead and pick that one. Okay, you might not be right, but it's just that common. Okay, so we use them for everything. They're enzymes. We use it to help contract our muscles. I can't even go into all the depths of what different proteins we have in our body. Important, there's going to be a nitrogen group on our proteins. Because when we talk about the urinary system, we're eliminating nitrogenous waste. A lot of it is come from the breakdown of proteins. And when we break down a protein, what is the smallest monomer of a protein? It's an amino acid. So the monomer of a carbohydrate is glucose. The monomer of a fat is going to be fatty acids and glycerol. The monomer of a protein is amino acids. When you see the term peptide, you should think protein because it's still a covalent bond, right? We are in the organic group, but we're going to call that covalent bond a peptide bond. So if you ever see the term peptide, think protein. The type of protein just depends on what's attached to the amino acid. So you're going to put some amino acids together, but there's going to be an R group on there, which means that there's something additional added onto it right there. So they get linked together as peptide by a peptide bond, which is the name of the covalent bond. You're all humans, but you got your own name. Okay, so here's an amino acid, here's an amino acid. These are the monomers of a protein. You put two of them together by a peptide bond, boom, you have a dipeptide. These things get really big, and so sometimes they'll be in different sort of shapes. If they're all just straight line, that's going to be primary. But when you see something coiled, that's going to be a secondary. Or if it's a pleated protein, then it's going to be a secondary protein. Tertiary and quaternary is when they get all wadded up. Obviously, quaternary is going to be quite a mess, but it's how two or more polypeptides wad up. So here's a primary structure. It's just amino acid, amino acid, amino acid, amino acid. This is the coil. So a secondary structure. This is a pleat. They're trying to show with color change, but of course this is a two-dimensional picture of a three-dimensional object, so it's hard to see.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
A lot of times as we move forward in A&P, we're going to use the term a globular protein or a fibrous protein. A fibrous protein is more like a strand. A globular protein is more like a glob, very compact, round-ish, and that's all you need to know about that right now. Okay, the term denaturing protein. When you break apart these tightly bound globs, globular proteins, or fibrous, but they're a little bit more stable. When you break them apart, they denature. So when you, let's go big. When you've got a steak and you add some salt to it to tenderize it, you're denaturing some of that protein to make it more tender, to make that steak more tender. When you cook it, especially if you like it well done, you have completely broken apart those proteins. And that is irreversible, right? So, for example, you cannot undo cooking an egg. And then enzymes, I've already mentioned that they are the catalyst in a lot of our biological reactions. And I've mentioned that they're proteins. They're globular proteins. They increase the speed of a chemical reaction. And important to note that they don't get used up. They'll migrate on and go help another reaction. They can be used over and over and over and over and over again. Enzymes are very, very specific. So lactase breaks down lactose. Maltase breaks down maltose. Acetylcholinesterase breaks down acetylcholine. You get the idea. Enzymes usually end in an ASE. So if you see ASE, you should think enzyme. Because they are so helpful, not only do they speed up the reaction, but less energy is required to get that reaction going. So they decrease the amount of activation energy required for the reaction. So this is showing how much energy would be required to get this reaction going all the way up here before you finally get the product. And then on this one, if you add an enzyme, it doesn't take nearly as much energy. And I believe I've used the example of, if you drink milk and you are lactose intolerant, are you going to poop out milk? No, it's going to be broken up. But it's hard for your body to do it if you don't have lactase. So maybe you get a bellyache or something because you're using a lot of energy to get that lactose broken up. This is showing the specificity also. This amino acid, this amino acid fits specifically into this hole of the enzyme. If this was different shaped from this, they wouldn't bind. So maltase, if this is maltase and this is lactose, these two aren't going to fit together. So it can't do the job. Final category of organics, nucleic acids. So the ones that I've mentioned already, DNA, RNA, but there's a third one. ATP is a nucleic acid. It is very specific what its nitrogen base is. It's adenosine. It's much smaller also than DNA and RNA. ATP is smaller than DNA and RNA. All of these guys can get broken down into a monomer called a nucleotide. So nucleic acid is the large molecule, and nucleotide is the small molecule. These guys are actually the largest molecules in our body. DNA is our genetic blueprint. Super important, it gets housed in the center of the nucleus, very protected. It is a double helix, and listen, it is made of a deoxyribose sugar, so there's that pentose sugar. It's made of a phosphate group, and then it has a nitrogen base. That's its nucleotide. There's 4 different nitrogen bases that you're going to find in DNA. Adenine, guanine, cytosine, and thymine. Those are the 4 different nitrogen bases that you can find. And A and G, adenine and guanine, are purines. C and T, cytosine and thymine, are pyrimidines. So the purines and the pyrimidines bind together by a hydrogen bond. It's called complementary base pairing. Like this. So the DNA, if you uncoiled it, it would look like a ladder. The sides of the ladder are made of that pentose sugar and a phosphate, pentose sugar and a phosphate, pentose sugar and a phosphate, pentose sugar and a phosphate. Then those A's, C's, T's, and G's are in the middle, right here, sticking out in the middle. Again, the purines pair with the pyrimidines and they bind together by a hydrogen bond. RNA is a little bit different from DNA in that it's single-stranded instead of double-stranded. Sometimes you'll find an RNA inside the nucleus, but he does most of his work outside of the nucleus. His pentose sugar, instead of being deoxyribose, it's ribose, but still has the ribose sugar and the phosphate, the ribose sugar and the phosphate, the ribose sugar and the phosphate. And then instead of having uracil, it has thymine. Instead of having thymine, it has uracil. So there's different kinds of RNAs, and when we start making proteins, you're going to meet them. There's a messenger RNA, a transfer RNA, and a ribosomal RNA. Okay, ATP also has the pentose sugar. It's going to be a ribose. It's going to have a phosphate group, but its nitrogen base is going to be adenine. It's actually going to be adenosine, so it's an adenine containing nitrogen base. There's a little bit more to it. But what happens, the way that we get energy from this guy is very simple. Do you remember I say when you break a bond, you release energy? He's the major player of the kind of energy that we need to stay alive. What will happen is if I say adenosine triphosphate, if I cleave off one of those phosphate groups and make it adenosine diphosphate, when I broke that bond between two of the phosphates, I have released the kind of energy that makes us stay alive. Losing just a phosphate group creates the kind of energy that we use to stay alive. Here's the picture of it. So this is adenine, and then we got this little extra thing right here, adenosine, so what? And then the ribose sugar, and then the 3 phosphate groups. Take off one of those phosphate groups, we have ADP, and lots of energy. This is examples of different ways that ATP keeps us alive, but truly you just can't even, you can't summarize on a slide all the different things that ATP does for us. So I'm hoping you don't feel too scared of chemistry. It was pretty biology based. Thanks. Bye.